For the last 30 years, we've had the ability to diagnose inherited genetic disorders in human embryos, what's called pre-implantation genetic testing. And this has proven to be a really useful approach for patients who are at high risk of passing on a particular inherited disorder to their children. It's really given them a very valuable reproductive option that avoids pregnancy termination and helps them to have unaffected children. It's been a bit frustrating though that in all those years we've been able to do these diagnostics, but we've never had any ability to actually cure an embryo. That means that the embryos we find to be abnormal simply have to be discarded. It would be very attractive to us, the concept that maybe we could detect an abnormality but then fix it and have that embryo still available for transfer to the patient. That was all science fiction for many years. But recently there have been great advances in what's known as genome editing technologies. These are molecular tools that allow us to very precisely change pieces of the human genome. So for example, if a patient had a mutant gene that was causing a disease in the family and they wanted to avoid transmitting it to the next generation, you could think about potentially using these tools to correct that defective gene, maybe in the embryos they produce during IVF. Now, that's all very controversial. People are worried about this, and with some good reason. The safety of these kinds of approaches really has not been fully assessed. While we always describe these methods as being very precise, you're just changing this defective gene, in reality, there's the possibility that some of these methods could change other areas of the genome which were not intended. With luck, this will not be the case, but that needs to be fully established before this is ever really utilized clinically. And of course, there's ethical questions about, do we really want to do this kind of thing to the human genome? We talk about doing this in embryos. Why do it in embryos? Why not just help adults or children who have a defective gene? Well, the reason why it's attractive to think about doing it at these embryonic stages is that it's much easier to correct the gene in all of the cells. You can imagine an adult is made of trillions of cells, and many of them are deeply inside the body. So there is a difficulty in accessing those cells to correct the genetic defect. If we have an embryo that we've made using IVF, it's just sitting in a dish in the laboratory. All of its cells are right there, and so correcting the defect in all the cells, and therefore in all the cells of the body that will be produced, is much easier. So that's the attraction of doing it there. It's a double-edged sword though. The other thing that you get if you do that is that you will, whether you wanted to or not, also alter the gene in the germ cells, in other words, the sperm or the egg. And that means that the alteration you have made will be passed on to future generations. So any change that you induce will be stuck with humanity for the rest of time, potentially. And of course, this makes some people a little bit worried. Now, you could argue on the purely medical side that this is a good thing. Not only have you cured the defect in the individual who will come from that embryo, but actually they don't need to worry about that particular inherited disorder for future generations of their family either. It's gone. But of course there are counter arguments that people sometimes don't feel comfortable with that level of intervention. So these are discussions that we still have to have. We need to nail down the safety. Is it safe? How is it best to do this efficiently? And do we really want to do this? Depending on the results of those experiments and those discussions, perhaps it may be very interesting to correct these defects in these embryos, but we're not at that point yet. Of course, we're all aware that there have been reports out of China about the actual clinical application of these procedures, and I think that's kind of been almost universally condemned by the scientific community and the medical community at the moment. Clearly, it's not ready for this sort of application yet, and irresponsible to do so. Here within EVRMA, we've remained very interested though in this as a research tool. and also the possibilities it may offer in the future if safety and ethics can be resolved.